All right, we are going to cover today some of the technical challenge of launching and operating biomarker-based screening programs. So I'm really excited to be here today with Will, who's a computer scientist by training, but has spent a good part of the last decade thinking about running large-scale clinical research programs that are focused on basically biomarker-intensive clinical trials. So we're talking about not just genetics, but also other forms of omics. So Will, he studied at the University of Cambridge and Wellcome Sanger Institute. That's how we met. And I think he's going to have an interesting perspective to bring to you all today about what it means to run these kind of programs in maybe thinking about it from first principles in a computer scientist's perspective to make them scalable, to make them reliable, robust. So Will, I don't know if you want to say a couple words about how you got interested in clinical research, and then we can jump into it.
Hey, yeah, I'll just briefly say thanks, Patrick. Great to chat to you all today. As Patrick mentions, I've been a computer scientist by training, so I'm a mathematician as well. But as you mentioned, Patrick, it has been almost a decade that I've been thinking about these sorts of things, which is wild to think about. So I'm looking forward to going into a bit more detail later on. And yeah, great to be here.
Great. So I'm going to give a quick intro here and just set the scene of what we mean when we say complex biomarker screening. And I think actually the past decade is a good backdrop of this because a lot has changed. So what I mean when I say this is using genetics or other kinds of omics or digital biomarkers. That could be things like an MRI or other kinds of measures to separate patients into treatment arms based on something that is measured by that biomarker. So this example here is an example from an oncology program that shows treatment based on the genetic variant or tumor underlying a particular tumor. But this can be applied to every form of genetic or even non-genetic medicine, and think about how we move to a world where treatments are more precise and more tailored to the individual. So why is this so important right now? If we look in 2005, only 1 out of every 20 medicines that was approved by the FDA had a biomarker and could be considered a personalized medicine. If you fast forward to 2018, it's more than 40%, and today it's likely to be over half that are approved every year that could be considered a precision medicine. You can see on the right in this figure, the number of approvals of personalized medicines continues to grow. So for those of you in the audience that are working on clinical research studies, there's a 50-50 chance if you're at the trial stage that you live in this world today. But in the next couple of years, it's only going to grow. So the vast majority of people on the call will work on these kind of biomarker intensive programs within your career. And why has this shift happened? So a number of companies have looked retrospectively at their pipelines. You can see AstraZeneca, as well as a broad comparison to papers on the right here that includes data from GSK and a number of others, that using genetic biomarkers in particular increases success rate in trials dramatically. So we know that failure rate is upwards of 90%, but using a target that's genetically validated can push this to be two to three times more successful. So this has pushed a lot of investment into early research and development, and that's over the years has moved into clinical trials and clinical development. And why is this, just briefly, the case? So genetics in particular can tell us a lot about whether a drug is likely to be safe. And the useful thought experiment here is to say, if we know that that the increased level of a particular gene or protein causes a disease, and then what you might want to do with a drug is knock that protein down and bring it back to normal levels. But a fair question to ask is, would that be safe? What happens if we knock the gene down too much, or if we knock it down in the wrong tissue? And it turns out that genetics is a really useful way to look at safety through the lens of what we might call human knockout studies. So you can look for people that actually have the gene naturally knocked out. They don't have the disease. And you can ask the question of what else happens to them. Do they have, are they healthy? Do they have other comorbidities? And this helps you to get a picture of, if we were to knock the gene down using a drug, would it be likely to have off-target safety effects? And what can genetic data tell us about that in this real-world experience? The other important use of it is determining the targets to go after in the first place. So using genome-wide association studies and something called a phenome-wide association studies, you can look at which genes are linked to the progression or risk of a disease, or also subtypes of disease. And this is probably the the case that you're most familiar with, and you use genetics as an anchor to better treat a target population. And then finally, these can be used in novel trial designs. So you may be familiar with the concept of a master protocol that I'll come back to a little bit later, but basket trials and umbrella trials, like the one that I showed you a couple slides before, that use a genetic marker to, or other kind of biomarker to more efficiently assign patients to the right treatment or the right arm of a trial, are becoming more and more popular because they allow us to run these kind of complex studies in a much more efficient manner and basically pre-screen patients, determine which of a basket of potential trials or potential therapies they might be eligible for or might benefit from, and then allocate more efficiently into trial arms. So we'll come back to these concepts, but this is why this phenomenon is occurring. One of the challenges is that running these kinds of programs requires a large number of people to have already been tested. So if you're looking for a subset of patients with a common disease like Parkinson's or Alzheimer's that have a genetic profile or have a particular genetic profile, then you're relying often on the fact that they've already been tested. But you have this chicken and egg problem where the healthcare system won't pay for testing until there's an approved drug, but it's hard to get drugs approved until you find patients with testing. And so the result is you have this logjam where testing isn't available in the healthcare system because there's no evidence that it helps patients, but you can't generate the evidence without testing. And many of you will be familiar with this, but one of the solutions to this is to coordinate a number of different organizations to do recruitment, genetic testing, counseling, and break this vicious cycle to start to generate the evidence by running large-scale biomarker studies, including genetic testing and others. So that's a little bit what brings us today to where we are today. But some of the challenges with this approach is that if you're running one of these kinds of programs, you probably know that most biomarkers, especially if they're new ones or haven't been studied for longer than a decade, they're going to be absent from healthcare site and recruitment databases. So any estimates that you make for feasibility are going to be really challenging to do. But also, when it comes to recruitment, you have to generate this kind of data from scratch because it's not in the healthcare system. And as a result of that, you have very high screen failure rates. So it can be in the upwards of 90+% in some of the diseases we work on, because if the genetic variant itself is only present in 10% of the population, and then you layer on all of the other exclusion criteria, you can have an incredibly high screen failure rate, which makes these studies very difficult to conduct because sites often can't screen that many patients, or you get site fatigue, or it just simply becomes too expensive to screen that many patients. And these are a couple of examples where you routinely see really high screen failure rates. And again, the need for pre-screening is important here. You also have, from both the patient's perspective and the drug developer's perspective, a lot of duplication of efforts. If you've got a bunch of different trials that are just treating one drug in one group of patients, then you have a lot of duplication of efforts, particularly around recruitment, where you may be simultaneously spending on a bunch of different campaigns that are ultimately going for the same group of patients. And then the last two here are about country and site selection. So if you're looking for a specific set of patients with a particular genetic biomarker, the site selection and country mix that you go for may be very different based on the genetic epidemiology of that disease and where patients who are affected by that particular genetic subtype of disease are more likely to live. So for example, with PNPLA3, this NASH genetic marker in point 2A up here, this is a lot more common in people of Hispanic ancestry, and in particular certain Latin American ancestries. So you may choose the countries and the sites that you operate on based on density of patients, that not just patients with NASH, but patients that have NASH and have a particular ethical origin that makes them more likely to have the variant of interest. And then finally, point 5 here is you may be able to tackle this in some cases by a simple brute force approach, but this makes it really challenging to prepare the market. If assuming you have a companion diagnostic or a novel biomarker that you're planning to bring to market, you want to make sure that doctors understand what this test is and why it's important, so that you don't go through all the effort of getting the drug approved only to not have doctors or patients understand that it exists. The approach that we've seen work in this, and the framework we've kind of built to apply to this, is if we think about the multi-step process of getting a new drug through to through to approval and to patients ultimately, we often think of a couple of big buckets here. So R&D, early preclinical work, observational studies like natural history studies are especially important in rare disease, and then moving into clinical development. But rather than running these in isolation, you can think about running them as an integrated process where the same patients that are part of the natural history study run here may indeed go on to be part of clinical studies later down the line. And also patients who are maybe genetically tested in this part of the process could be recontacted post-market to let them know about a drug that may be effective for them, or via their doctor to let them know about a drug that may be effective for them. So we're not going to spend too much time going into the details of setting up these patient finding programs and the technology. We're going to spend more time on the technology behind the virtual waiting room and the screening process, but we do have a number of materials on our website that can give you more info about these patient finding protocols and how to go about setting those up. So just before I turn over to Will here, this probably reminds you of this master protocol concept if you've come across it before. And this is a trial design paradigm where you profile a biomarker, whether it's genetics or otherwise, and you match patients into the right investigational therapy depending on that biomarker. And this has been very commonly used in oncology, but it's gaining momentum in other diseases as well. And this pre-screening approach very naturally aligns with this master protocol approach, where you're essentially doing the same thing, but through a real-world evidence pre-screening type of program. So I'm going to just introduce this here as an example of a early version of a real-life patient journey through one of these pre-screening programs. And I just want to make the point here that it's incredibly complex. There's a lot of steps that a patient or healthcare provider or combination could go through in one of these. And what we're going to talk about today is how to build these complex flows from the ground up using a modular and scalable framework that helps you to think about, instead of starting with this big mess of a wiring diagram, how do we start from really basic building blocks and then build up from there to get to something that's measurable, robust, reliable, compliant, and all the things we look for.
I think this diagram here encapsulates, I think, part of the point that we are trying to make here. These programs can be complex and can require multiple touch points from participants who are involved in the process, but not just them, sites, HCPs, and many other processes that run asynchronously. And if you've got thousands of participants running through this process, you have really a lot of pragmatic challenges that you have to face and you have to find solutions for to keep these programs running, and to ultimately manage the relationships of the people who are who you're trying to serve as a group of people. So the really the investment, or the way to think about this, is that if you can keep these people engaged in the program and you build these relationships, suddenly you're not just building a base for your later clinical research, but you're also incentivizing and building a community around what you're trying to produce and the therapy that you're looking to try and produce anyway. So I'll just outline a few of the sort of concrete challenges that the industry really faces when trying to conduct sort of large-scale international programs like this. These are sort of the following. So there are, one is finding a central source of truth for how many participants are in whatever steps. So if you ask the question to yourself, how many people are dropping out at a certain stage? If you're looking to coordinate 3 or 4 different organizations and passing the baton, as it were, to these organizations, it can become increasingly difficult to know what the source of truth is and where the leaks in your funnel are. So this is one of the really big challenges that is faced by managing this process across a network of organizations, is just figuring out what the source of truth is. And if you don't know the source of truth, suddenly you're flying blind in managing this big process, and it can be really, really challenging. Another really challenging piece is ensuring compliance. So if you're operating not just in the UK, but in the US and in European countries as well, and you have people going through this process, you have data being collected in different jurisdictions, there's always a concern about what the regulations are in these jurisdictions, and how do you build one system that makes sure it complies with wherever it's operating. And increasingly, if you're looking for to deploy an international therapy, not just in a kind of a standard market like the US, really understanding how to bring a program like this and ensure you're complying with all the regulations that are there is a big headache. So another challenge that is very frequently faced. Likewise, sort of as mentioned before, this is really a long-term engagement play. So if you are trying to build relationships with thousands of participants who are running through this program, there's no point with generating this upfront cost and then having only 10% of them come back in a year's time. How do you actually give these people a really great experience to so that they tell their networks and to get excited about the research program and the therapy that you're actually trying to generate? So really, this is a core challenge that you face in managing these processes. Likewise, relating to point two, if you're working in different jurisdictions, we really, you need to ensure that you're working with as diverse an audience as possible, so that you're not just creating a therapy for a particular population. So to really effectively bring this to the world, you need to be able to run these sort of patient finding programs in whatever languages that you need that these people speak. So if you're just doing this in the UK, or in English, you are really not catering for a huge fraction of people who'll be potential users of whatever therapy you're making. And likewise, finally, just to mention on, you can think of versioned study releases. So if you are operating a study with a set of materials that have been approved and you need to run a protocol amendment, how do you actually instigate that change and propagate that to whatever jurisdiction that you're working in, right? How do you make sure that data that you've collected in previous versions of the study is actually compatible with data that's generated in later versions of the study? How do you make sure that all of that process is auditable, such that if you're if you are audited yourself, that you have a clear trail of what data has been collected under what purpose? So really, the way that what I'll introduce later on enables all of these sort of key challenges and actually provide solutions to all of these. So with that, I'll tee up some of the ideas that we use at Sano to really enable you to sort of address these challenges and to address them head on, without just without having to find solutions at a late date. So I think just to sort of bring back the quite complex map, one of these study processes that Patrick shared earlier, if we sort of zoom in on the left-hand side in this corner with a few of these boxes, and really these boxes are a well-defined step in a participant's experience, whether they're being asked a question, whether they're being shown an information panel, whether they're being passed to, introduced to a research site, for example. What we've done as a company really is to try and figure out what reusable concepts can we use, or to sort of at a basic level, what is going on here, and can we sort of define some concepts that we can then reuse later down the line, and enable this entire process to be really, really granular, clear, and easily traceable. So there's a few concepts that I'll sort of introduce to you that we'll see, and I'll explain how this relates to this complex map here. But really, from an abstract point of view, really what we try and do is provide very, very clear and well-defined steps in a user's experience, whether that's obtaining e-consent, whether that's asking them a question, whether that's giving them an information panel or an opportunity to request a DNA kit. So these comprise the blocks of the steps in the process that we see in this map. And what we can also define is this essentially a flow, which really, the way that we think about it is an ordering or a sequence of these blocks in whatever order you want to put them in. And it means that in a given sort of experience for a participant, we know exactly what they're going to experience at a certain point in time. So really, these are sort of two core concepts that we use at Sano to build out these myriad very complex digital experiences, but the core concepts are actually very, very simple, we hope, at least. So just some examples of what we mean by a block. So each of these, if you imagine that back to that complex map earlier, what we mean to say by each of those boxes is really kind of just like a page that a participant will see, right? Whether it's giving their simple consent to an information panel here, but whether it might be adding their digital signature to a block. We've got blocks that actually enable that functionality, and I'll talk about compliance a bit later on. But these can be also very simple, such that just asking the user what their full name is. We can put these blocks in whatever sequence we need to give the experience later down the line, and what's required for the program. Likewise with just adding multi-select questions, asking them about a variety of different diseases, whether it's diabetes or something else. This can all be customized as well. So these are some blocks that we can show and add to this program. And you might be thinking as well, a flow is not just a linear selection of our, if I'm thinking about my protocol, this is a complex set of things that might happen. So within a flow, we can branch these blocks depending on answers that a participant has given in a previous block. So these can be essentially as arbitrarily as complex as you want them to be. However, the key product challenge for us is also making them as simple as they can be, because you need to keep things as simple as possible. But the point is that no matter how complex these protocols are, there is always a way that it can be represented within this framework. And likewise, we can sort of branch and compose these flows together such that whatever requirement is necessary for a program, of which there's a myriad set of requirements, we can actually map this to the sort of framework that we develop to then make things very, very easy to get a lot of things for free later on down the line. We can sort of map these programs to very, very well-defined, to a configuration of very, very well-defined core concepts. So just to sort of follow on from this, we can start to think about, well, this is one of the ways that we at Sano think about, what it is to join a study. What does that actually mean, right? We can actually start to align here. And so really we can think about a pre-screening flow that might have what we have here. This is actually a success criteria, essentially, that we restrict users from completing a flow unless they meet a certain criteria, right? So, sort of a natural concept to, get your head around. And, from that point, we can prevent a user from actually completing a flow in the way that will make them join a study. And so we can restrict the number of people actually joining. So suddenly the concept of joining a study and being in this program or being in this state is very, very well defined. And, once users are part of a study itself, they might be able to join other flows. And we also have a wide variety of different types of activities that they might participate in within a given flow, which I'll go into. The other thing that happens in these large-scale, long-term programs is asynchronous steps, right? So unfortunately, we don't live in a world where, you can submit a sample, put it in the post, it gets to the lab and is sequenced all in the same browsing session, okay? Unfortunately, we don't live in that world. I think we never will be unless we have these chips sort of in our computers. So there's a whole process of asynchronous steps, which often need to happen before we can then bring users back into the process and allow them to continue. So one of these is the process to order a DNA kit. So this is a DNA kit ordering block that we can add into the process. All the functionality that's involved here is modularized such that it's all included when you include this block in your study process. Participants would receive a kit in the mail, and they are put in the process and then re-engaged back into the platform a bit later on. So I'll just go into a bit more detail here. So this is what we mean by these awaiting steps. So within a given flow, we might not have all the information, or the participant might not have all the information that is required to complete this flow. Perhaps the labs need to receive the kit, the QC needs to be completed, but all of these steps act independently and receive communications from whatever is required here. So if a user comes back to the site and asks themselves, what's going on with this study, what's the process, we will actually be able to tell them exactly where they are in that sort of in that whole process, in that flow. And when we receive the the information to bring them to the next step, we will re-engage them, and they can see exactly where they are. So all of that complexity is handled. The whole relationship management is handled by this system, which is very well defined, and it has a number of benefits that I'll go into a bit later on about, with analysis and compliance and auditing as well. So this is hopefully this gives you, I mean, some of an idea of what, this really, what the way that we think about this, quite complex study design process or process map really is about decomposing this into a set of flows that can be joined together. And it enables us to keep a handle on this complexity and make sure we have all the analysis built in so that we know exactly how many people are at each stage. We can re-engage people as is necessary, and not have to build a separate website or a separate system for each part. We can reason about these things within the same framework and the same language. So now, just a sort of switch tactic, I mean, we really, as study process managers, we think a lot about the holistic view about a study, but to a participant, all they care about is where they are and where's next, and also why should I continue to care about your program. So we spend a lot of time on the product side, to really try and make this experience, an A+ experience for the participants who are, in these research programs. So we provide really this sort of customizable dashboard view where a participant can understand where they are in the process with these, as we call them, cards, essentially, that can be, we can program and ensure that the right cards are shown to the right people at the right time. So for this person, they are being sent a kit right now according to their card, and they can actually start the process to claim their kit, submit the sample, and continue the flow with this call to action here. So that, of course, that only will happen once they've got the kit. We've also depending on what stage they are in the process, we can provide them with sort of relevant content to keep them engaged as well, and customize this depending on a whole myriad of set of things about that particular participant. And as sort of mentioned before, a big part of this process is on asynchronous comms. So it's about how do we keep these people engaged for a long period of time. So really we address this by great email comms, and as is one of the ways that we do it, but also by concierge service, where we would actually generate and have actual chats with users on the phone and via SMSs as well. But I think this is one of our strengths, is that our brand, our content, and our design really allows us to hit a really high average open rate for these emails to keep people engaged in the process. So we regularly see open rates of 65% versus sort of an industry average of 21%. And if you can just imagine that delta, if you have 1,000 people running through a process where they have to come back to a certain site, once they've received a test, that delta can really affect how many people you get at the end of the funnel. If every single time you're losing 80% of people. So this is something that we really try and optimize for. We take very, very seriously, and we have sort of design, product engagement teams as well to optimize this and make sure that we are at the cutting edge, and making sure that we can do this better than anyone else in our industry. And just to show a few more of the types of cards and comms that you see going through, we try and make these as minimal and clean as possible, but also to programs. And so as an ongoing engagement piece, this is just a few more examples about the types of experiences that we look to give to these users. So for example, there's a customizable dashboard section with updates around the study to build a sense of community around what's going on. So instead of just sending a kit back in the mail and not having any idea about what's happening, we really want to make sure these people in these programs are part of a community and feel that sense of that it's not just them a part of it, because we know from experience, and I think just intuitively, that if you're part of a big cohort, you're just much more incentivized to stick to the end and actually you be receptive to the really to the asks later down the line. There's nothing worse than putting your kit in the mail and a year later having someone ask you to do something if you've not heard from them for a year. So we really just want to make sure this is avoided, and we can give people a continuous great experience, such that for these longitudinal programs, you can really reap the rewards later in the process, as opposed to be stuck with a big problem later in the process. There's a few pieces that I think talked about in not too much detail, so I'll go through those now. So one is around regulatory compliance. So this is a big headache for a number of the people that we work with, and I think one of the benefits that we can bring is ensuring that we can actually have properties attached to each of these blocks. So we can say, well, this ethnicity block is only valid to be used in the UK, or this particular block implements the recommended ethnicity questionnaire set as recommended by the FDA, for example. So we've actually given a large set of sort of target markets. We've mapped all of the best ways of asking these types of questions to make sure that you're not having to scramble and understand what the cultural expectations are of running these sorts of questions and these sorts of programs, or what to expect. So we've really done a lot of the hard lifting to make sure it's compatible in a wide range of jurisdictions. Medical research regulations such as GCP, we're actually obliged to retain a certain set of that information after a user has gone past a certain point. So what is part of the sort of product development cycle, figuring out that fine line between the rights of individuals versus the responsibilities of a company conducting an international medical research program. It's something that comes out of the box with this process. So that's one of the big benefits, that many of these anxieties are not present if you run programs within this framework that we've made. And just finally, with I touched on this earlier, but there's a variety of sorts of e-consent. So in the US, there's 21 CFR Part 11 compliance, and in Europe as well, EIDAS is obviously stipulates certain types of consent about and what is not valid, and we've got blocks that actually comply to those regulations and can be used at any circumstances as well. So depending on whether you want to run in a program in the US or the UK, you can actually customize. It's the experience of deploying that program is actually just as simple as dragging the right block and using it in the right circumstance. And that's part of what we try and provide, or that clarity about what to do where, and make it a matter of just choosing what to do, as opposed to figuring out what the right way to ask the question is. And just lastly, I think just on translation, and this is a screenshot from an example study page, which is our sort of hub for the programs that we run. It's absolutely vital these days that we can make these programs as accessible as possible, working not just in popular, I think the most populous countries, or commonly run research programs in for example the US or the UK, which may be multilingual as well, but also in countries where English is not widely spoken. So what we've built this system in a way that we can actually ensure a master copy of the study content, but actually integrate with translation partners to customize and to actually translate all of what's required in the study, and populate it for users who are in whatever jurisdiction is necessary. And we've built that in a way that is very effective for us, easy for us to do and manage, but also very easy to version, such that if you want to, or if you need to, version a specific or release a new version of a study, according to an IB amendment of whatever it is, we can actually translate the differences that are required and propagate those differences to different jurisdictions where the program is running. So this is another part of core part of what we do, and it doesn't mean you have to run different programs with different companies in different countries. You can have a sole provider with a sole source of truth about what's going on in every jurisdiction that you operate. So this is an important consideration as well. Okay, so I'm just going to go through some of the points that I listed earlier, and the intention was to sort of answer many of these questions. So just sort of touch, now that we sort of have been introduced to these concepts, now we can actually start to really understand and have funnel analytics essentially for free. So given the framework, if the study process map is implemented in that way, essentially you get analytics for free. So you can know exactly which stage everybody is at. You can understand where people are dropping out, and we can actually attach automated communications and triggers to wherever we want in the process, which can be, scheduled and customized as necessary, which is really, really powerful. The other thing is around ensuring compliance. So I won't talk about that anymore because we had a whole slide on that. Another piece is around long-term engagement. There's a lot of content there that I spoke about, translation as well, and also these versioned study releases, which I've touched upon previously, but I think should not be underestimated. I think one of the key considerations is now that every single component of a study is thought about in a very sort of concrete and well-defined way, we can actually think about an entire, in the same way that you might version software, you might version a study release. And the benefits of that are when you actually look to analyze a program, you have absolute security and guarantees about what materials have been shown at whatever stage of the program. So if you're concerned about an audit trail or understanding what whether you're compliant with the ethics approvals that you've received, you can instantly have that reassurance essentially. And you can also reason about IRB amendments in a much more well-defined way. So these things should be avoided, but oftentimes you do need to release a new version of a study and submit it for ethics. And if you can actually think about these things in concrete concepts, suddenly that becomes very, very easy when previously it might require months and months and months and huge teams to organize. We can do that with the actual framework and within software extremely easily. So I'll leave it there and leave some time for Q&A, but hopefully that gives you a flavor of some of the tools that we use to make these complex things much, much easier, and to manage these things, giving many of the benefits that I've talked about now. So I'll just open the chat now, but I'll leave it there and perhaps pass back to Patrick and we can go into to Q&A.
No, thanks. There are a couple of questions that have come through that I'll play back to you. Is a good one, two really good ones, and then I've got another one that isn't in the chat, but that came through, I think, directly to me. So the first one is, if a clinical study is targeting an age range that is not technology minded, do you think that the process might actually increase dropout rate? I think it's a good opportunity to talk about phone support and other ways of making sure it's not a web only world. I think it's a really great question.
This is something that I didn't talk about here, but it's a really important thing to address. So in every single program that we've run, we still think about the actual digital experience as what the source of truth is. But for each one of these blocks, we actually provide phone support such that an MSL, so a medical science liaison, or someone on the phone can actually talk directly to whatever participant is involved in the program and submit answers on their behalf. So as you mentioned, there may be circumstances where a digital only experience is not the best medium to maintain these relationships, but we also provide this support layer such that a participant may only actually interact with us as a program via someone over the phone, and that's totally fine. That's an additional layer that we provide as part of a program, but the actual core data then gets fed through into the same system with the same analytics and the same versioning and core concepts. So hopefully that answers your question.
Yeah, thanks for that great question. I can take this next one. So there was a two-part question about genetics, and first off, if you have a mutation that might be linked to a disease but the participant is asymptomatic, what does that process look like? How long might you need to wait for a treatment? How long is data retained? And then there's a related question about how you think about how genetic variants are correlated with the disease when sequencing the sample. I think it's a really good question, one we think about a lot. For everyone's benefit, there's a concept in genetics called penetrance, which is basically the answer to the question, if you have the gene variant, do you necessarily get the disease? And if something is highly penetrant, it means having the gene means you're definitely going to get the disease. But if it's incompletely penetrant, then having the gene might increase your risk, but it doesn't mean you're going to get it. And so that's a factor to consider, whether or not the gene is fully penetrant, but it also matters whether the participant is diagnosed with the disease today, or whether they maybe carry a gene that says they're definitely going to get it, or they're very likely to get it, but they're not actually diagnosed today. So when it comes to designing these studies, those are really critical pieces that feed into the strategy around communication of participants, data retention, some of these points you mentioned. So it's hard to give an answer about how we'd handle every possible approach, but one of the trickiest ones is a study where you may be testing for something that's highly penetrant, meaning if you have the gene, you're very likely to get the disease, but the person doesn't have the disease today. This is a really challenging one because you may be delivering news that's really psychologically challenging for participants. So one of the things using Will's framework that he showed here that you would want to consider here is something like a pre-test counseling session as being mandatory in a study like that, where you maybe can't take part in a study like that until you've spoken to a genetic counselor that explains the potential outcomes before someone makes that decision. But in a different program where maybe somebody's already diagnosed and the genetic variant is going to tell them about potential eligibility for a trial that they're going to discuss with their doctor, but it doesn't really change their treatment, then you may say that in that circumstance, genetic counseling may not be mandatory beforehand, but it is mandatory afterwards, for example. So you can use those and basically codify them into this framework. And in terms of data retention, it's a lot driven by regulations. So under GDPR, there's a maximum data retention period, but also more importantly than that is about clear communication with participants around what the data retention actually is for a study. And that's normally captured in the informed consent. Great. We have time for, I think, one more question, which was about some of the differences between regulations in different parts of the world. So maybe you can share a few examples around what would change in these kind of programs between, say, the EU and the US or the EU and the UK. What are some of the kinds of pieces of this framework that might need to change as you think about running in different countries? We've got language, obviously, but what else are the important things to think about?
Yeah, it's a good one. This is where it can get very complicated, actually. So I think some of the good examples, as we talked about in translation, but the actual parts in the process which need to be different. So for example, in France, you need to be with an HCP in order to return the results of genetic testing. So if you were to sort of build that into the system, a very different, a coordination between the HCP providers and the patient and the participants needs to be sort of made available, which funnels information into the wider study process maps so that you understand and have a source of truth about what's going on at any point in time. Another example is with e-consent. So in different European countries, you have different requirements about what digital signatures is acceptable. So in some countries, actually, there's a requirement for physical signatures and actually integrating with the postal service to send signatures, verify that they exist, and actually integrate that as part of the process. Becomes more and more complicated. Of course, some blocks are very easy to work with. Other blocks are more challenging. So we can actually analyze we can have a view on how successful these processes are, but to work in some countries, there's a wide variety of different process specifications that are required that we can actually model and include as blocks, and then can be used in and configured in these studies. So those are a couple of examples.
Yeah, those are great examples. Thanks everyone for sticking with us and for the great questions. Thank you, Will, for the really clear overview. I think it was a good summary of some of the challenges of not doing this right, but also I think this framework can be applied to almost every one of these kind of complex programs. So thank you. Appreciate you taking the time to do it.
No problem.
All right, everybody else, enjoy your day, and we'll see you next time.
Thanks, everyone.